There's been a lot of disease pressure this year in both Alberta and Saskatchewan, and actually Manitoba for that matter. And everybody that has sent in germinations this year so far has found that there's been a lot of disease, mainly of Fusarium, Septoria, Pyranothera, root rot, and those types of things. Now, all of these diseases can be managed very well with seed treatment, but Ted very kindly asked me to come today. This is actually the first presentation I'm making this year on disease, and I'm really pleased to be here because I'd like to get the message out that through some of the conversations that we've been having on the telephone about some of the problems and some of the concerns over the diseases. So Mitch has said we're fully accredited. Our main services include seed quality, and through that it's germination, vigor, and purity. Seed health is a big component of what we do, and that's either done with the traditional plate or molecular analysis. We're the first private company to be accredited to do hybrid crop inspection, and we also have a large interest in doing research. We're located now in Nisku, Lethbridge, and Winnipeg, and we were the first commercial lab to receive accreditation back in 1989. In addition to being 9001-2008 certified, we are also internationally accredited and have been for the last 3 years, along with our Canadian Food Inspection Agency accreditation, and shortly we'll be working on becoming ISO 17025 because we've just partnered with a company in Sweden to offer a broader testing for varietal identification and molecular analysis in genetics. So the seed-borne pathogen testing, this is traditionally how we do our analysis. On the right-hand side, so on your left, that's a sample of barley, and then on the other side, on your right, those are peas. And for the longest time, that's how we did our traditional disease analysis, through putting seed that's been sterilized with a bleach solution onto a plate that's known to promote that pathogen. So these are just typical plates that we would use. They contain potato dextrose, which is a starch. And we've also used things like V8 juice. Blackleg, for example, that's found on canola, does very well on V8 juice. So there's certain components that we use to promote the growth of those pathogens and then look at those colonies. But more recently, we've got into molecular analysis, and we are doing all of our club root testing with molecular and a lot of our DNA work on Fusarium graminearum as well. So this year, actually this graph represents the last 5 years of Fusarium infection on both wheat and barley in Alberta. And you've been given a little handout that gives you the specifics on the number of samples and percentages of infection that we've been getting. But you can see how, because of drier years up until this year, Fusarium graminearum, both on wheat and barley, was fairly flat. But most diseases are very weather dependent, and we've had perfect conditions to promote those pathogens this year. Not just with Fusarium, and as I said, root rot, but even things like ergot and sclerotinia are very much present in our cereal and canola samples. Now, this shows you 2011 versus 2012. In 2011, our average infection of positive samples for Fusarium was sitting at 0.75. This year, it's doubled to 1.5. And we're seeing anything from 0.5 of an infection, which is 1 seed in 200, all the way up to as much as 12%. And the reason for this is because of the year that we've had. And just about all of our samples that we've got into the lab in the Lethbridge area particularly have had some infection. This shows you the percentage of samples that are testing positive with the DNA and the plate. And I'd just like to take a few minutes here to just discuss this. On DNA, we typically find more infection. And the reason for that is because obviously with DNA, we're able to find the spores whether they're dead or alive. On the DNA plate, it's only the live spores that we're able to detect. Now, the DNA test does a soak of the seed and then a grind. So we're keeping the sample intact. So the sample that comes into the lab for testing maintains its integrity. It doesn't change from the time that it comes into the lab and is tested. So if there are live or dead spores there, we will find them because of the sensitivity of the DNA test. Now, when we do a plate test to follow up with a positive DNA, we'll often find that it's negative. And the reason for that is that we found a low-level percentage of infection in the DNA, but the plate, because we're only testing 200 seeds, and those seeds have been disinfected with a bleach solution, if the spores were on the seed, so they were just on the surface, and they weren't systemic, then that bleach can actually wash out those spores off and they don't show up in the plate. So I can just explain for a minute, we've had a lot of complaints this year because the pressure is so high, the incident level of finding disease this year is so much higher than it's been in the past. We've been criticized for finding it on DNA and then not finding it on the plate. And I know we need to get out and educate our clients more about the difference between those two tests. Now, the good news, if you find it on DNA and you don't on the plate, means that the chances are that those spores are actually dead and they're not going to cause a problem. But the problem is that we are reporting that we have found a very low level of infection on our report. And because Fusarium is controlled under the Pest Control Act, that is an issue when it's on the report, because we are reporting that we've found it. But we haven't actually reported that we've got a percentage. So if you follow where I'm going with that, it's good risk management to know if you're finding it on the DNA. So this is diseases that we would typically see on cereals, and I've got some fantastic live material here that I'm going to show you in a little bit. So the pathogenic fungi, Cochleobolus sativus, which is root rot, Fusarium graminearum, which leads to head blight, Fusarium species such as seedling blight, the roots and the crown rots and head blight, and Pyranothera, which is a complex group that make up the net blotch and tan spots and leaf stripe, and then of course Septoria, which is Septoria nodorum, which we typically only see on wheat. And then the saprophytic or weak pathogens, and these are generally here all the time, are Alternaria, Cladosporium, and Epicoccum. And these are really quite aggressive if we find mechanical damage on our samples, and I'll talk to you a little bit about that when we get to the live part. And then there's the storage molds like Aspergillus and Penicillium, and of course Penicillium is your bread mold, and we often see that when seed has been stored with a high moisture content. And they just affect the surface, spoil the appearance of the seed. Now, this particular group of pathogens and saprophytes, we do a fungal screen on this, and I'll talk to you a little bit about that. We've actually got two special tests that give you a diagnostic profile of all the diseases that you would commonly find on your samples that are submitted and give you the perfect match of seed treatment to work with these specific pathogens. The 4 main pathogens that we see are Septoria, Pyranothera, Fusarium, and Cochleobolus sativus. Now, I want to talk to you about Fusarium and my opinion on why it's spreading and how we can manage it. Now, you can see here Fusarium in the last year. So this represents our testing season currently. The blue line, Cochleobolus sativus, very flat. We don't typically see root rot in wheat. We pick it up, but we don't typically consider it to be problematic. Pyranothera, yes. Septoria, yes. And Fusarium, definitely. So let's start with Fusarium because it's the one that's causing the most concern. Now, Fusarium is a parasite that's capable of affecting more than one part of the plant, which is why it's particularly problematic. It not only affects the root and the crown, but it affects the head as well. And then it's a parasite as well as a saprophyte. So as a saprophyte, the spores are living on the stubble from the previous crop, or they can survive in the grasses around surrounding the field. Now, because it's a parasite that affects different parts of the plant, there are different ways that we can control it. And when we see it in the laboratory on the seed, we offer our clients the choice of having it retested again with a treatment. And we find that the treatment is especially helpful and works extremely well to give us a better interpretation of germination. We do realize that this is not a fix everything, and we're certainly not promoting it on seed that doesn't have good germination and vigor, but we are definitely promoting seed treatment as a means of managing it on your seed because it gives early protection to those susceptible or sensitive seedlings. Now, also Fusarium, I don't know if you know this, but as I said, it affects the crown and the root, and that's typically where we see it in the laboratory, on the roots and on the crown, and sometimes on the leaf itself, and obviously on the seed as well. So that disease then, when it's growing in the field, if you see it at that stage, very seldom goes on to becoming Fusarium head blight. Fusarium head blight is caused by the spores from the previous crop or from spores that are sitting surrounding in another field that could potentially have had it, or in headland grasses. And there are a lot of hosts for Fusarium in the grass family, so it's important to keep those headlands mowed down prior to flowering before they head out, because that's when Fusarium head blight becomes an issue. So those spores that are sitting on the stubble, or even for that matter, as I say, in surrounding areas, become ascospores, which are wind-blown, or they become coinidia, which are water-splashed. So this year we've had 2 very important elements that have contributed towards the spread of Fusarium across the province. We've known that it's been in Manitoba since 1994. It was found in Saskatchewan in 1998. And in 1999, Alberta declared it a pest. And we've had a very strong management program since then. And here we are, 13 years later, doing very, very well. We have not got the issues that Saskatchewan or Manitoba have, partly because we don't have the weather patterns that they do, but this year, coincidentally, we did. Now, I've always gone out and said, Yes, it's very weather dependent. It has to be warm and it has to be wet. But I thought, I say that all the time. So I started to study some papers that I could find on Fusarium. And I don't know if any of you have seen this, but I picked up a paper from a study that was done just recently in Brazil on Fusarium graminearum. Very, very interesting. They did cloud spore collection in several fields and had the plates that I showed you placed around the field and in different parts of the field. And they did 2 measurements, one at 9 o'clock in the morning and another one at 9 o'clock at night. And they found that the most activity was at night when the humidity was at 30 to 50%. That's when they picked up most of those spores, which I thought was very, very interesting, because under irrigation, you have that sort of greenhouse effect. Even under dry land farming, we've had those humid conditions this year, lots of untimely rains, and perfect timing when those spores can be either wind blown, we've had a lot of wind this year, or rain splashed onto the head, which is when they start to germinate into the endosperm and cause those issues. Part of this study too was that the only other time that they found as many spores collected was during the day when it rained. So it had to be over 80% humidity, and that was the only time they actually found equal number of spores. And I just thought, I've often heard in Manitoba that part of the reason they've got such a high incidence of Fusarium there is because they have humid evenings and the temperature doesn't fall much below 20 degrees. So that's a big reason why we're seeing quite a bit more of a spread. And the other reason is that not that many people are practicing no-till. And of course, when you're not ploughing old crop residue down and burying it, that's another source for infection to live. And of course, it's also a soil-borne infection, and that's where treatment initially on the seed is very, very important. And the only other one is the Pinophyra, and those are the net blotch, and leaf blotch, and tan spots. And they tend to behave quite a little bit like Ascochyta in peas, because they develop pycnidia on their leaves, and they're spread by rain. So when it's raining, we'll see quite a bit of a spread. Same kind of thing for barley, exactly the same diseases, but here you see root rot, definitely quite problematic because it affects barley. Barley is very susceptible, and the Pyronophyra as well. This is the disease profile on pulses. And again, not only are our cereals affected this year, but also our pulses, and that goes for peas, beans, and chickpeas. And typically what we've been seeing is Ascochyta, which is leaf blight and the root rot complex. And this is the one that you get the Ascochyta affecting it underneath the canopy of the leaves. It creates sort of like a white mildew. You end up with a dark circle full of pycnidia. When it rains, it spreads onto the pods and then onto the seed. And the problem with this one is that it affects the point at which the seed attaches to the seedling. So it's a problem because obviously the seed is a food reserve for the seedlings, and without that, no growth can occur. And then pretty much after that, we've seen a little bit of Fusarium, lots of sclerotinia, lots and lots and lots of sclerotinia this year, and Stemphillium, which is the leaf blight that we see occasionally on chickpeas. This year, we're seeing 40% of our samples, 40 to 45% of our samples affected with true loose smut. And this is a real issue this year. That's more than we've seen in years past. And true loose smut, as you know, affects the endosperm or embryo in the head. It's a disease that starts as a mushroom type of thing, and it basically permeates into the seed and creates little veins into which are actually the disease into the seed, and you can't see them inside. You have to do an embryo extraction and then use a dye to be able to find the smut. And we've had an extreme number really this year in our barley. So the highlights this year, I've pretty much said that increased incidence of low-level Fusarium east of Highway 2. We're seeing club root spreading from different areas, and our positive test results for Fusarium this year are running at 30% on the DNA and 25% on the plates, respectively. We do, as analysts, have to be aware of pathogens because we're not just looking at things like mechanical damage, frost damage, glyphosate damage, those types of things. We've got to be very aware that there are pathogens that can cause problems. But we can't say when we open up the blotter definitively what it is without having Dustin, our pathologist, identify it correctly. Now, once we find it, we realize that we want to go back to the customer and let them know. They've not asked for this, by the way. When they ask for a germination, they're just asking for a germination. A lot of labs just give them the germination. They don't give them any other information. And I feel that's very important that we tell our customers what's going on with their seed so that they can make the best decisions possible as early as possible too. So we not only give a diagnostic profile on whether the seed, when we've done a germination, is dead, abnormal, dormant or fresh, because any one of those things alone or in combination can mean that that seed has been exposed to something, plus something of a pathogen screen. We don't conduct the fungal screen without obviously getting their permission, but if we find pathogens on the sample, we feel it's important that we let them know, and we can continue on advising them throughout the course of the season. So seed-borne pathogens, I don't have to tell you this, do reduce plant establishment and crop stand. Some of them are more yield robbing and have more economical value than others. And for us, knowing that you've got pathogens and knowing about the health of your seed is very, very important so that you can use the appropriate seed treatments.